All right, so our last speaker for today is Samuel, and he comes from the DAMP lab, and he's going to talk to us about a different service than anything we've heard about so far today, which is the microfluidics fabrication services through the DAMP
lab. All right. So hello, everyone. Good afternoon. So welcome to the last presentation of the day. So we're switching gears here to a different type of automation, right? So we're not talking about robotics anymore. Now it's microfluidics. But we're yet on the same theme of like, how can we make automated like frameworks for designing and fabricating the not biologicals, now microfluidic devices. All right, so then we're gonna be visiting this building shortly in 10 to 15 minutes, and then the microfluidics fabrication is located on the 6th floor. And it looks like that. So we have an office space which we share with Cider Lab, and then there's a prototyping lab, fab space, and scope room. And that kind of like summarizes how do we do. So we design things, we fabricate, we assemble, and then we test, right? So basically that. And then here, just to give a little context of how the microfluidics fabrication started, as everything in the DAMP lab came from foundations of Professor Doug Densmore lab. So the Cider is his primary research group here. So Doug has created, like, developed many software and hardware, and everything towards like automating the framework, as we mentioned, for designing or rapidly designing and fabricating, in this case, reliable microfluidics. So we got like a bunch of tools here, not only microfluidics related, just to mention a few of them. You can always check on Doug's website, on Research Lab website, Cider. And there's also hardware, a lot of different hardware here. And then I mentioned some of the areas that we like, which are droplet generators, sorters, and screening. So you're going to see some videos about how we came up with different like components, like, or primary components for us to start assembling like more complicated devices. So electronic embedded microfluidics, we're going to discuss a little bit about VLSI, for very large scale integrated microfluidics, which is a different vision that we got in the lab, and then also some capillary based microfluidics. So we've done all of that, and I say we because I was until last year I was Doug's postdoc working for the Cider Lab. Nowadays I have like my own research lab, which is sort of like starting right now this year. But then as the microfluidics manager, of course, I've been always talking about this with all the staff that we have, which are basically undergrads working with us. So in terms of areas that we explored there, so again, talking about that vision, how do we automate the design and fabrication? We usually start with some software tool that help us figure out what is the best geometries for this microfluidic device. And then this one specifically is a machine learning based software that can, from user-defined performances of a future device, it figures like what is the best geometries that can achieve that performance. And then you can always also like couple that with semiconductors or some, in this case, conductive ink that will have like electrodes, and then will integrate with electronics later on. And then finally, you got this like very nice device. Now, of course, it's not that easy to get a very pretty like device like that. Of course, there's like many iterations, but then that's the beauty of this type of framework, like rapid prototyping. So we use the type of like techniques that we use and materials that we use allow us to fail fast and just move on until we get something like that, like this in the matters of like a week. Another area that I was talking before that we're pushing a lot now recently is the VLSI. So those are complicated devices. It's hard even like to see sometimes. So those are mainly for routing multi like fluidics. If you have needs for valving and pumping different liquids and mixing them up, so you gotta like design something like that. Now this type of device is differently from droplets. Yes, it's a multi-layer device. It requires what we call control and flow layers, and it's hard to design two different designs at the same time because you need them to be completely aligned, right? So then there's software tools like 3DUF that came also from Doug's lab that we can help us. It's a CAD tool that help us design that with precision. So you don't have to worry so much about what's going on in the control flow layer. You just make them all together, and you got these pretty devices in the end. Right. So then, just to give an overview of the fabrication, not with the purpose of explaining every single step, but mostly just to show you how from multiple steps like that, we can decrease the time, and then we're putting those metrics up here. So how fast can we fail those devices? So we say designing and milling takes about like 1 hour and a couple of dollars, like $5, and then just keep going like cleaning and adding those electrodes that we mentioned, conducting ink ones, device assembly and setting up and testing. So everything takes like about like a day if you think about, and then a couple of dollars. So of course, if you can always see those like a summary to compare with photolithography, for example. And then this is what we would call like the gold standard method that takes days, perhaps weeks, and thousands of money, right? So then, of course, there's a compromise. Like you can gain and you can lose. And then sometimes what we lose when we do the rapid prototyping is just the roughness of the surfaces and the size of the features. So you have to take that into consideration. But depending on what you need, you don't need so much precision. You just need something to just like proof of concept and move on, right? And that's kind of like our take in this environment. And just to give you a little idea of like what type of microfluidics they look, some of them look like that. Those are a bunch of components and devices that have been published throughout maybe the last 4 to 5 years. And then just to give you an idea, so this is like impedance sensor or capacitance sensing, a sorter, a merging of droplets, and those are for droplets, microfluidics, right? And then here's like pico injecting, and you see a little injection going on here, and you can inject something that will fluoresce after you culture. Like if you just keep those droplets containing like fluorescence, and you expect some reaction to occur at 37 degrees, for example, you can incubate those droplets and then wait for a response. So like I said here, and then you can think of all different ways of merging different concentrations of different liquids and things like that, and then having those droplets being generated as well as we show here, but then with the single cell encapsulation capability, if you want. And then, of course, the purpose of like, why do we need a droplet in any case, right? Or in general, like what we're trying to do here is just making tons of those droplets, each one representing one experiment that you can run. Like thinking about different wells in a next 96 or 384 well plate. Now we're talking about thousands and thousands of independent experiments that you can have in the droplet, right? And then you can just keep generating them and experimenting them after cultivating them. Now, another thing I want to bring, what's going on in the microfluidics facility related to the VLSI. Now we're studying large community of cells in there, and then we're making devices like that look like that one. We call this multi-layer device or motherboard, and with the vision that we could have multiple communities being communicated with this motherboard that valves and routes signals underneath those communities that sit down here. Now, for this purpose, what we want to bring the attention is we can CAD design them, we can fabricate them relatively fast, and we can have software that can help us control those those platforms afterwards. So all of that, something that we can do in the dev lab. And then I want to bring also the attention to the outreach opportunities that we also form inside this facility. So this one specifically, it's an IGM team, and it's a theme of this year that we're competing. And those students are the ones making those devices, getting trained and having fun with us. So this is also a very good educational program. Yeah, so almost the last slide here. So this is like about the industry and academic collaboration that we're currently having. And again, a facility that can help us prototype something fast, something that is relatively complicated, could be easily prototyped, so we can give directions to a more complex device later on. So this is a collaboration that is being funded by BioMade in collaboration with Professor Rabia here from the Electrical Computer Engineering Department. And we are the ones having fun with students, undergrads, making those funny microfluidic devices to test commercially available electrodes. So we're doing the easy job so we can get it complicated later on. But then probably at this point, we're going to have proven some concepts and then given this to the folks here at BU. Then so that's my second to last slide about microfluidic consortium. So that's another thing that we're pushing a lot in the microfluidics facilities, just being out there, right? So we're engaging with the community, just like we're doing here. In the microfluidics, we do the same. So last year, we organized a microfluidics consortium open day that happened here in Boston. This year, we were involved as well. We always bring those folks to tour in our labs, and it's always a good way to stay connected to everybody, create sponsorships opportunities, maybe engage with the industry and so on. If you want to know more about that, of course, you can always follow that consortium on LinkedIn, and then you're going to be more than welcome in the community. And then this is my, kind of last slide. So industry engagement. So we are not at the stage as like CTL and Dave was just talking about. So we're not providing services. Like we don't have off-the-shelf services and full PDFs to say, just come and grab your like microfluidic device. What we do differently is we just talk. So if you have something, something to be tested like relatively quick, we can be your folks like testing them out. And then the way is just making contact. And then just like I presented, like the other industry engagement that we got, it's like through contacts and communication, talking, and then we say how we can provide something for you that takes perhaps 6 months, 12 months or something. So everything kind of like takes time for us yet, but then we're launching this probably next year when we're going to try to, shape some better format for this type of link engagement. Right now it's just basically on demand. I have my last slide here. I just want to give a shout out to the DAMP Microfluidics, which is me and an undergrad right now. Yash is doing a great job. Of course, there's many people from the CIDR lab that should be mentioned here. And of course, the entire DAMP lab. So happy to take questions.
Could you give an example of what project somebody might come to you for as a service, so you can get an idea of what they might be requesting of us?
Yeah, I've been thinking about like screening antibiotics, for example, is something that I can imagine, like something that requires droplets, like tons of droplets, and you want to test different concentrations of some antibiotics against a specific sample. I don't know, it could be bacteria or some other organism. I think that would be a nice way to test. It could be even through the VLSI or even droplets, like I said, controlling peak injection. So creating this sort of like platforms for screening different candidates for targets is something that comes to my mind right now. Yeah.
I'll just add, just really quick. You might be like, I'm confused. Everything was going well. This became a very end of the day. Why are we talking about microfluidics? If you remember the way I said the DAMP lab started, I said I made software. People said this was cool, but I have no lab to do with what it came out. Similarly, the same software that my lab makes that makes DNA, it also makes a graph how the DNA should interact in a microfluidic. So now I need to connect to a plate to make microfluidics. So I need to do 2 connections. So we were already offering this microfluidic fabrication process for our group. So I said to Samuel, why don't you make it available to anyone that's using the DAMP lab anyways? They're already coming to us. That's the genesis of it. And the software you would pick, kind of Lynn's question, is you literally say, I want to make a droplet, I want to incubate it, I want to sort it. You plop pieces down on a canvas, you say, this is what I want, then you say build, and it goes to Samuel. So they may come in with an application, but you might just be very device-based and say, that's what I want to make. And so that's why this is coming up, because this is something that's very low-hanging fruit for us to already offer.